Hello, and thank you all for joining us today for this Celltheon webinar hosted by Samba Scientific. My name is Jason Amsbaugh, and I'll be the moderator today. Our speaker today is Divya Goel, Vice President of Business Development at Celltheon. A bioprocess engineer by training, Divya oversees platform development and technology licensing for Celltheon, as well as working with the technical staff to evaluate new bioprocessing technologies and develop efficient workflows that can be leveraged by the greater pharmaceutical industry. A couple housekeeping notes. First off, if you have a question during the presentation, please submit it into the Q&A box in your Zoom panel here. And that way we'll have a queue essentially, and we'll get to questions and answers after Divya's presentation. Additionally, this webinar will be recorded. So if you miss any part of it or would like to rewatch it or share it with colleagues, you'll be notified a few days after the webinar when it's available for on-demand viewing. Before we begin, we have just one quick poll for the audience. You should see it launch on your screen here. And really just one question. What brought you here today? Are you interested in cell line development services, production services? Are you interested in platform licensing options? Or are you just here to learn? So please go ahead and click all that apply. Thank you for your responses. And with that, I'll go ahead and turn our time over to Divya.
Thank you, Jason, for the introduction, and thank you, everybody, for being here today and joining us for this webinar. The presentation title today is Foolproof Biotherapeutics Manufacturing for All Proteins. So just to get started at an overview, we'll initially be talking about a little bit about Celltheon and our background, and then jumping into what we see as current challenges in biotherapeutics development and how we feel that that needs to be addressed by developing a robust preclinical platform. And some of the solutions that we've come up with in our technology integration have been the Supercell CHO-K1 cell line, our OmniCHO transient system, our Goldilocks transposase, and our ChromaSelect cell screening platform. And I'll end with a couple concluding remarks. So at a high level, Celltheon is located in the San Francisco Bay Area in Union City, California. We provide cell line development services and also other services for the greater biotherapeutics market. So that includes protein therapeutics, platform technologies and services for cell and gene therapy and vaccines as well. But the topic and focus of today's presentation will be more for protein therapeutics. Our facility here in the US provides end-to-end product development for manufacturing of protein therapeutics. We have a long-standing track record of delivering over 10,000 proteins to date since our inception in 2012. And we are a full service CRO. We provide cell line development and protein expression services, assay development, and PD services. So since our inception in 2012, we've worked on a number of complex and challenging molecules over the past 11 years. And there are three main challenges that we currently are seeing in biotherapeutics development and manufacturing. So the first is that we've observed an increasing trend in development of what we consider more complex biologics. Monoclonal antibodies were considered a gold standard and were more common just within the last couple decades, but we've been seeing increasing number of bispecific antibodies, fusion proteins, DARTs, BiTEs, scFvs, and probodies that have been starting to make their way into early stage of preclinical development and are now advancing into the clinic. And so these individual molecules are obviously a lot more potent. They're significantly engineered. And while they are clinically more beneficial, they do come with their own unique developability and manufacturability challenges. So for example, with bispecific antibodies, the challenge is always to optimize the expression of the heterodimer variant as opposed to the homodimer that is secreted by the cells. And if that ratio is not optimal, then we observe low yields in the downstream purification. With certain types of fusion proteins, we observe issues such as truncation and aggregation. And with molecules like SCFEs and probodies, we also have observed issues with truncation and low titers. So these next generation therapeutics have these unique developability and manufacturability challenges. And what we've observed is that each specific program requires customization every single time a new particular challenging molecule or challenging critical quality attribute needs to be optimized. And that's not ideal because we obviously increase the timelines as we start to induce more customization into the development process. Second, kind of age-old question that we've seen since the beginning of biomanufacturing is to meet the annual demand for a therapeutic, do you scale up, scale out, or scale in place? So scaling up, of course, we're familiar with the extremely large 20,000 liter fermenters, which have been around for decades and are no longer necessarily needed for most monoclonal antibodies as titers have been increasing over the past few decades. But as these more complex and challenging molecules are coming online, we've seen the same issues of low titer, low yields in the downstream. So we do need to scale up in some cases to meet the annual demand. Another trend that we've seen increasing over the past several years has been to scale out, and that's through introduction of something like a 6-pack of 2,000 liter scale bioreactors. So this way we are minimizing the risk by not having to run one single 20,000 liter run. We can spread the risk out over multiple batch productions at a smaller scale. But the issue with that is still that you have increasing cost of goods, increased cost of labor for running each individual batch at a smaller scale, and overall increased CapEx and OpEx. So the ideal answer here is, of course, to scale in place. If we can address the manufacturability and productivity challenges in the manufacturing platform itself, it allows us to stay at a much smaller volume, and therefore each individual batch, if lost, is less of a risk and less of a cost burden. So that would be the ideal scenario. And the last issue that we've seen with biotherapeutic manufacturing is that the classical development workflow seems to be incredibly siloed. So you start with your development with the discovery process, and then you do typically some sort of a variant screening, developability studies, and that is what we consider early preclinical development. Now, this stage of development typically relies on transient transfections, and we see these groups increasingly using things like transient CHO-S kits or HEK293 kits. And then from there, variants are moved into the formal cell line development stage once we've selected our top candidate for development. And there's a transition that happens from our transient cell line to our stable cell line. And that typically tends to be some sort of a CHO-K1 based cell line. So there is not only a very siloed sort of a development structure where you have an antibody development group using one platform and then having cell line development using a different manufacturing system. Then from there, it gets tech transferred into process development, and they do further process optimization, media screening, et cetera. And that can further change the critical quality attributes. So what we're seeing here is that the development and early screening is based on certain assumptions and the critical quality attributes of the cell lines that are available at the early stages of development. And so this disjointed nature and handoff from various stages in the development process can lead to high risk, especially when we're developing these complex biologics, which require critical quality attributes to be built in from the beginning of the program. So how do we see the best way forward to foolproof your biomanufacturing is basically addressing these three topics: that there must be technology platform integration from early discovery through late stage of commercial manufacturing. There must be manufacturing efficiency. Even for complex biologics, we must obtain higher titers and better yields in the downstream. And we must be able to build in critical quality attributes, screening or optimization into the manufacturing platform to minimize the requirements for customization for each individual program, which just further extends the timelines. And our answer to that is the updates and developments we've made in the Celltheon Smart Expression platform, which I'll be talking about today in each of these individual technology components. But at a high level, the Celltheon Smart platform, we really wanted to make sure that it was robust and holistic so it could offer a solution for that technology platform integration issue that I mentioned earlier. So at the top of this technology stack in the development lifecycle, we have our supercell host cell line, which is a CHO-K1 derived host cell line that we've optimized for certain unique critical quality attribute expression. So the advantage of using the Celltheon Smart platform is that we've developed two systems that can enable early discovery through late stage manufacturing using our same host supercell CHO-K1 cell line. So our OmniCHO transient transfection kit is developed using the supercell CHO-K1 cell line. So it allows antibody discovery and groups to essentially use the CHO-K1 cell line that you would eventually use in manufacturing, but at that earlier stage of antibody development, discovery, screening, etc. And so from there, we can obtain a robust data set and really predict the critical quality attributes and optimize the variance for what we would like to achieve for that particular therapeutic program. And then from there, advance our lead candidates into later stages of stable pool generation using our Goldilocks transposase technology. So in conjunction, again, with the supercell CHO-K1 host cell line and our high titer Goldilocks transposase technology, we can then create pools on our top variants, and then from there select one variant, which would move into a more formal cell line development. And from that point, once we've selected our top candidate, if there needs to be further optimization of critical quality attributes, we can use our ChromaSelect cell screening platform, which allows us to screen 100 million clones for unique critical quality attributes and really select out the ideal CQAs at a very early stage in cell line development, and then proceed into the later stages of process development with the Celltheon Smart Bioprocessing platform. And the Celltheon Smart Bioprocessing platform leverages commercially available media and feeds for a 14-day platformed production process. And we've tech transferred the system over the past several years to various different CDMOs. We've used different bioreactors in the scale-up process. So our system has worked with 100% success rate in scale-up from our 3-liter bench scale system to much larger scale subs for clinical manufacturing. And so our system is essentially agnostic to which platform you decide to use for manufacturing. So a little bit about the top level of our system, which is our CHO cell line. So with conventional CHO cells, when you transfect in your gene of interest into the cells, they're folded by these classical protein folding chaperones. But what we see with difficult to express proteins is that they're secreted in a misfolded state. And that induces the unfolded protein response and induces ER stress on the cells. And there's a feedback loop that happens, which reduces the production of the nascent polypeptides. And you tend to get an overall reduction of the expression of these difficult to express proteins. And overall, what you tend to see is much higher levels of aggregated material of the protein that is being secreted. But with the supercell platform, we've developed a novel CHO-K1 derived progeny that is not genetically modified, and it has upregulated enzymes that are related to protein folding and redox maintenance. So that enables expression of very difficult to express proteins. So the enzymes act on these classical protein chaperones, which are endogenous to the CHO-K1 cell line, but enhance the protein folding of these difficult to express proteins. So we see higher levels, higher titers of these DEPs and more monomeric forms of the molecules. And here's just a list or a subset of some of the molecules that we've expressed in the supercell platform successfully. Proteins like lipoproteins, bispecific antibodies, nanobodies, cytokine fusions, and even very high molecular weight proteins up to 1 milladalton have been successfully expressed in our system. And a couple other attributes of the supercell platform are that the product quality are, as I mentioned, we have high levels of monomeric species produced by the cells. Very easy scale up, has an 18-hour doubling time, no issues with clumping, low oxygen consumption, and very high specific productivity with our platformed fed batch production process using commercial media and feed. We obtain peak cell densities of 15 million cells per ml, very high titers of 8 to 12 grams per liter, and very high expression of difficult to express molecules. And lastly, the metabolic profiles of our CHOK1 cell line is optimal as well. We observe low glucose consumption, low ammonia, and low lactate. And on the left-hand side here, you can see the lactate profile for 13 unique clones that we profiled during the fed batch production run. Cells peak at about 2 grams per liter of lactate, and then that's subsequently consumed by the cells. And similarly, ammonia production peaks at no more than 4 or 5 millimolar. And we've also confirmed scalability of our system in several different culture formats, and several CDMOs have worked with our platform as well and have confirmed scalability in 200 and 500 liter single-use bioreactors and greater. This is a data set that we generated in-house, comparing expression of an antibody at a shake flask, 3-liter stir tank bioreactor, a 20-liter wave, and a 50-liter stir tank single-use bioreactor. And we can see very consistent cell growth profiles as well as expression level at harvest. So as I mentioned earlier, we developed the supercell as the top level, or the base rather, of our production system. And on top of that, we've built several other platforms that enable our tools for things like drug discovery and antibody screening. And one such platform is the OmniCho transient transfection kit, which has certain unique attributes in using the supercell as its base host cell line. So some key features here are that we typically observe about 6 to 10-fold improvement in titer for standard monoclonal antibodies, and over 20-fold increase in titer for what we would consider difficult to express proteins. So I'll draw your attention here to this graph on the right-hand side. And here we are showing a comparison of expression data at our day 12 harvest time point in the OmniCho system versus the day 12 fed batch production system of an industry-leading transient protein expression platform. Here we have a variety of different benchmark proteins and monoclonal antibodies. And we can see with the industry-leading platform, titers are quite good, about 500 mg per liter to about 1 gram per liter. But in our OmniCho system, you can see a significant increase in expression, increasing even up to 4.5 grams per liter, and several other molecules which are 6-fold to 10-fold higher expressors. So this is a subset of molecules we express that we consider to be well-behaved, good expressing molecules in the industry-leading platform. And we see substantial increases in our system. So the advantage of using this system is that you get significantly higher expression, higher titers with the OmniCho system, even for well-behaved molecules, significant improvements in expression. But there's a seamless platform in using our supercell host CHOK1 cell line at this early discovery stage. So you can do all of your analysis for developability, optimization, and your screening using our system. And then another advantage is that because the titers are so high, it is a very good platform for using for automated workflows, especially during the antibody discovery process. So if you needed to do 24 deep wall plate formats for library screening of antibodies, that's easily doable with our system using an automated workflow. And on the converse, it's also a highly scalable process. So unlike some other transient transfection kits, as you increase in scale, the titer tends to have a drop off. But with our system, the platform is incredibly scalable. So as you scale up to larger and larger culture volumes, larger vessels, you maintain the same level of productivity even at larger scales. So the previous data set was showing the expression level for several benchmark molecules that were well behaved. And in this table, we're showing the actual titers for a subset of molecules which are actually considered difficult to express, or in some cases, as you can see here, there was 0 expression in the industry-leading transient transfection kit. And these include molecules like several fusion proteins, bispecifics, membrane proteins. They're completely zero in the industry-leading platform. And in our OmniCho system, at least in some cases, we're getting some expression. In some cases, we are getting fairly decent expression. And at the end of this table here, there was some expression in the industry-leading platform, but in our system, we were getting exceptional titers of about 1 gram per liter, whereas in the industry-leading kit, we were getting about 100 mg per liter or less. And similarly, we have profiled the product quality of various different types of therapeutic antibodies in both the SDS page and size exclusion HPLC data set here, and we can see that overall product quality looks incredibly clean after just a single protein A purification step. So the next challenge that we wanted to address from the biotherapeutic workflow was the manufacturing efficiency. And that we've addressed using our Goldilocks transposase technology in conjunction with the SuperCell platform. Key features of our Goldilocks technology is that we're able to achieve high titers for all proteins. We are able to use a universal vector in signal sequence. So there's no optimization required at the start of the program. We just take the amino acid sequence, clone it into our gene of interest, and we express the protein of interest with the Goldilocks transposase, create pools, and there's no optimization required. Similarly, our cell line development platform or workflow is extremely simple and compatible with almost any CLD workflow. So there's no need for any sort of compulsory equipment. We have done even basic limiting dilution, subcloning, and looking at 50 clones, and we've been able to identify extremely high productivity clones. Furthermore, the Goldilocks transposase confers very high stability to the resulting clones and pools. And so that allows for a lot of flexibility in development. And overall, since it is a question that we are commonly asked, there's about 10 to 20 copies per cell. So not absurdly high, but somewhere kind of middle of the road. So this is a data set that we've generated for a stable pool, which was expressing at just about 7.5 grams per liter. We subcloned that by limiting dilution and obtained single clones. And we're seeing a nice distribution from the top 6 clones in the range of 9 to 10 grams per liter. So the other advantage of our system is that from pools, we can derive clones that are even higher expressing. So if we're looking at titers that are consistently in the range of about 8 to 12 grams per liter for most proteins, then one 2,000 liter batch would yield about 16 to 24 kilograms per batch. And that would be in some cases sufficient to meet annual demand for certain programs. So that really allows you to scale in place. And so a little bit more about the Goldilocks transposase and how we derive this. We screened 15 million transposase variants in a library screen and essentially narrowed it down to a top subset of variants, which we then combined mutations of, and then use that as our final Goldilocks transposase. And from that, we've derived a data set of expression for different types of molecules here. We have examples of an FC fusion protein, lipoprotein, some MABs, 4-chain bispecifics, and we can see a nice range just at the pool stage of about titers of 5 to 8 grams per liter. And here's some actual copy number data on some of these pools. So for single chain molecules, we can see about 11 to 20 copies per cell. And for MAB, the combined is about similar between the heavy and light chain, about 10 to 20 copies per cell. As I mentioned before, one of the significant improvements that we've made, and we actually intentionally screened for when developing the Goldilocks transposase, is ensuring that it conferred stability to the transgene. So here's an example data set of 3 of these stable pools that we created, and we passaged them in a 60 generation stability assessment without selective pressure during the whole 60 generations worth of passaging, assessed them in our standard platform fed batch process. And we see overall, there's extreme stability from generation 0 to generation 60 without selective pressure, just using this Goldilocks transposase element in our cell line development workflow. And furthermore, the CQAs between the pool and the clones are incredibly consistent. So here's another data set of a subcloning that we performed. with a pool that was about 5.2 grams per liter. And from there, we derived clones of about 7 to 8 grams per liter from top 6 clones. So when we evaluated the quality attributes of the clones versus the pool, by size exclusion HPLC, we see that they're all monomeric forms of the protein after a single protein A purification step. And then furthermore, by SDS page, again, just after protein A purification, we see very high levels of purity. And furthermore, just incredibly consistent product quality from pools to clones. And the last challenge that we wanted to address in the biomanufacturing workflow was to be able to build in critical quality attributes into our platform. And the way we've achieved that is implementation of our ChromaSelect system, which is a cell screening platform. The ChromaSelect system allows us to take a fraction of the protein of interest that would typically be solubly expressed and display it on the cell surface. And then we can screen by flow cytometry individual clones and sort them out. And we can screen for a number of different attributes, including titer, but also other quality attributes that are ideal for certain biotherapeutics, such as heterodimer aggregation levels, and then certain glycan species. And this is just a subset of some of the things we can screen for. And from there, we would sort more and more rare populations, which have our ideal CQAs, and we would plate them out into static cultures. And from there, we can continue with the cell line development workflow. The advantage of this ChromaSelect cell screening system is that we can screen 100 million clones within a 12-hour period. So it really doesn't affect the overall cell line development timeline. It's just like integrating an extra day into the subcloning process. And this is representative of what it would look like with the ChromaSelect platform. So here's a FACS plot. And what we would essentially do is take the more rare population here at the end tail and sort for these ideal CQAs before subcloning our stable pool. So for example, with the bispecific, we can screen for increased heterodimer formation. With the fusion protein, we can screen for reduced aggregation. And for things like probodies, we can screen for reduced truncation. And this is an example of some sorting that we did with a bispecific antibody pool. So the unsorted pool had a ratio of about 80% heterodimer to 20% homodimer. And after sorting the bispecific antibody pool, we increased the ratio to about 95% heterodimer to 5% homodimer. And then from there, we can interrogate individual clones during the cell subcloning process and further enrich for more levels of the heterodimer. And here is another example of a molecule that we sorted. It's a nanobody fusion protein. And in this particular instance, it is a bivalent nanobody fusion where the C-terminal region was prone to truncation or degradation, and that would render the molecule essentially non-valent. So we ran the molecule in a cation exchange HPLC assay. Region 1 listed here corresponds with the non-valent product, which accounts for about 27% of the product. The middle section 2 is the monovalent species, which accounts for 53%. And regions 3 and 4 are the intact bivalent species. So at this, before sorting, the nanobody fusion only had 20% intact bivalent product. And after doing the ChromaSelect cell sorting, we were able to increase the bivalent population to 51%. This is just at the pool stage. And again, we can then subclone and identify clones with even higher levels of the bivalent product. And lastly, this is an example of using the ChromaSelect cell sorting platform for enriching for titer. So the bulk pool here was just under 1 gram per liter. And essentially you can select out and sort out more and more rare populations. So we took 5% of the top population. And when we grew that up and performed a fed batch assay, the titer was just about 1.3 grams per liter. When we increased the population to about 1%, then the titer increased from there to about 1.5 grams per liter, and then the 0.05% increased to about 2 grams per liter. So by the end, from the bulk pool, we were able to enrich and increase the titer to about 2 grams per liter. So in totality, by being able to screen CQAs, titer, any sort of attributes, we can make incredibly undevelopable molecules into developable assets. And this is with no customization. This is a platform developable screening tool. So overall, the end-to-end cell line development timeline from initial gene synthesis, cloning, generating pools, screening single clones, doing clone evaluation, and then generating the final research cell bank with the top 3 clones is an end-to-end timeline of 14 weeks. So very rapid turnaround time from the start of the program through final tech transfer of the research cell bank for manufacturing. So in addition to providing the platform technologies from end-to-end drug discovery through manufacturing, we also provide auxiliary services for preclinical development. Some of these auxiliary services include upstream process development. So we can do process development in 3-liter and 50-liter scale bioreactors for process confirmation studies. In parallel with cell line development, we can also do downstream protein purification, including 3 to 4 column chromatography, process optimization, viral inactivation, formulation, and sterile filtration. On the bioproduction side, we routinely provide and support transient transfection services to support early antibody discovery workflows, all the way through material production for larger animal studies, anywhere from 2 mils to 50-liter scale. And we can also develop and produce material from pools and clones, purify them by 3 to 4 column chromatography, and provide low endotoxin material for animal studies. We also develop analytical assays, and we can tech transfer these methods as well. But these are typically assays that we would use in the clone selection process and to support upstream and downstream PD. So at the end of the development process with Celltheon, you receive a very robust tech transfer package, the research cell bank with a very high titer system, in addition to upstream and downstream processes and analytical assays, all developed within the same timeline. So in conclusion, we feel that a robust manufacturing platform should assist in early stages of drug developability and should make a drug manufacturable. It should handle all programs without platform technology customization, and this is especially relevant with all of these new difficult to express protein formats. It should stay reliable and cost-effective for decades. It should be easily scalable and transferable while maintaining critical quality attributes, and should be built on technology components that are leading edge and can continuously be updated with new advances, which we intend to continue to do. So with that, I will hand it back to Jason, and we will continue forward with the Q&A. Thank you so much.
Thank you, Divya. All right. So as a reminder, this webinar was recorded, and you will be receiving a notification when it's available for on-demand on-demand viewing. If you do have a question, please go ahead and click the Q&A button in your Zoom toolbar and submit your question. A couple here just to start. The first is, do you use mRNA for transposase transfections?
Yes, we do. We use mRNA for all of our transposase cell line development and transfections.
All right. Next, do you use any custom medias or feeds in your processes?
We do not. So we use only commercially available media and feeds for early cell line development all the way through the commercial production process.
Gotcha. Thank you. A question here, is your CHO cell line a GS knockout?
It is not a GS knockout. So it is just a derived from CHOK1, and we are agnostic to the selection marker that would be used.
Gotcha. Another question here, have you worked with bacterial cells?
We do a little bit of work with bacterial cells, but it's not our primary cell type that we work with.
Okay. A question here about the Goldilocks transposase. How does that compare to piggyback?
Yeah, so we've done screening with Goldilocks transposase versus the piggyback transposase, and we've seen consistently about a twofold increase in expression but with the Goldilocks transposase as compared to the piggyback. So that's been across all different types of molecules that we've tried, benchmark mAbs and difficult to express proteins.
Gotcha, thank you. Question, can you show a slide wherein glycan CQA is maintained across the scales?
Sure. So that is something we are working on, and we're happy to do a follow-up and share that data. We don't have it currently available in this slide deck, but I'm happy to provide that later.
Great, thank you. Are you guys looking into cell-free expression systems in the future?
Currently, no, not with this, for protein expression.
Okay. And then I guess it seems like this was actually addressed. Just asking if you'd made any bispecifics using your platform.
Yeah, so we've made several bispecifics with our platform. We've seen very good titers at pool and clone stage, 4 to 6 grams per liter just at the pool stage. And then from there, we can continue forward and do further subcloning and cell screening. And from there, we can increase titers even further.
Excellent. A question, is the Goldilocks transposase comparable with leap-in transposases as well? I think the question is comparable. Yes. All right.
So we have done a little bit of comparison with the leap-in, and our system also tends to outperform the leap-in transposase system. So we've done comparability with the leap-in, the piggyback, and overall our system still tends to be about twofold higher than both of those transposase systems.
Very good. A question here, I guess, on the business model. Is this a royalty-free platform?
Yes, this is a royalty-free platform.
Excellent. All right, a couple more questions here coming in. This question is, can your cells be lyophilized? But I think we're talking more about product here.
Yeah, products should be able to be lyophilized, the purified protein.
Gotcha. All right, let's give the questions here another minute and see if anybody has other things to submit. All right. With a transposase system, is it really necessary to select 100 million clones by chromoselect?
That's a great question. With well-behaved and a very high titer system like the Goldilocks transposase, for most molecules, no. Our intention for developing the chromoselect platform is really intended so that we can screen critical quality attributes. So Goldilocks definitely helps give us improvement in stability of the clones, as well as the overall titer, so the yields are higher. But the intention is that if you have a particular CQA that you'd like to enrich it for example, heterodimer ratio, it becomes an incredibly fast screening tool to be able to enrich for a particular CQA. So in that sense, the two in combination are incredibly powerful. For a typical monoclonal antibody, I wouldn't say it's necessary. But for more complex next-gen proteins, I would say it's an incredibly powerful combination.
Thank you. All right, let's give it just another minute and see if there's any additional questions. All right, well, looks like we're all done. If you do have any questions, please go ahead and email info@celltheon.com, and we can follow up with you individually. I'd like to thank all of you for attending today. I realize your time is valuable, and I'm glad that you spent it with us. With that, have a great day, everyone, and look forward to seeing you at the next webinar series. Bye-bye.
Thank you.